First of all, I want to introduce very shortly the Department of Neurology. We are not a research institution. We got every year around 2,500 patients, inpatients, and around 50,000 outpatients. We have altogether 100 beds, 60 active neurology beds and 40 rehabilitation beds. And our staff is around 150 people are working at the neurology department. 36 medical doctors and around 8-10 PhD students. So our main goal is to cure patients. What kind of patients do we cure? One type is the stroke. Half of the patients are stroke patients. In Hungary, we have around 40,000 stroke patients per year, which is the same as a small town. Half of them die, unfortunately. So it's a very high mortality. Parkinson's disease, I think all of you know Parkinson's disease, the shaking palsy. And concerning our distinguished guest, I just want to mention that one of an important drug, razagilin, it was originally, many of the chemical aspects was developed here in Szeged by Professor Bernard, Erhin co-workers, and Musa Yudim, the Technion. He did the big, great pharmacological job. Multiple sclerosis, we have around 8,000 multiple sclerosis patients. At our clinic, we have altogether 1,000, nearly 1,000, so we are quite big MS center. And again, if I think about the Weizmann Institute, copolymer was, as far as I know, made in the Weizmann Institute, which is a good drug for the treatment of multiple sclerosis. And also we have migraine patients. Hungary, we have around 1 million migraine patients. 10% of the population suffers from migraine. So this is really very important disease from the everyday practice. We have neuromuscular disorders like amyotrophic lateral sclerosis, ALS. So what I want to tell you that mainly we are involved deeply in the treatment of patients. And at last not but least, thinking again about our distinguished guest, in the last week I just met with Amos Korchin, a good friend of mine, and we organized together the next World Congress of Controversies of Neurology in Budapest. So it will cover all of the neurology fields. So coming back to the Department of Neurology, so what are the research activities? We mainly focus pathomechanisms, primary headache disorders, migraine, cluster headache. A very good collaboration with the Lund University. Lars Edvinsson is a key player. Neurodegeneration, Huntington's disease, with Flint Beale. That time he worked at Harvard, and now he's working in New York. Other stroke, cognitive impairment. And also we have a preclinical facilities, animal models of different disorders. And also we have some kind of drug development activities. I think in this stage I should thank to Professor Philip, who is an excellent organic chemist, deeply involved in the synthesizing of different compounds what we are investigating, and also to Professor Toldi. He's working at the natural faculty, and he's a very close collaborator for us. What kind of methods do we use? Histology, immunohistochemistry, molecular biology, genetics, neurochemistry, behavioral testing, and imaging. What kind of research groups do we have? On headache research group, neurodegenerative disorders, multiple sclerosis, neuroimaging research group. We have a relatively big biobank. We collected all of the CSF, cerebrospinal fluid samples during the last 20 years, and it is at -70, and diagnostic lab from human cerebrospinal fluid. And here is the CSF lab. We perform routine CSF measurements, cell counting, and also we use this material in the European network, which is a European bank network, and we involved in these activities too. Since neurology, as all of the medical discipline, is running to the individual-based medicine, personalized medicine, and we need biomarkers. That's the reason why we are investigating deeply this compound. Concerning the immunohistochemistry lab, we use, as I told to you, the migraine models and neurodegenerative models, and here is the neurochemical and molecular laboratory, HPLC equipment. We use determination of different proteins, and maybe we will get very soon a mass spectrometry from the Uppsala University. We get the promise that we will have this. And our student, Márton, is working basically in this lab together with Dénes Zádori. He's a resident, a very, very bright resident. He finished his medical studies. He started his training at the chemistry department after he joined to us. He finished his PhD thesis, and also he is now working at the bedside, but in the last year he got a quite distinguished award from the president of our academy. And our student is working very close to Dénes Zárdori. Concerning the neurogenetics laboratory, we were quite lucky because we got a EU support, the European Union support, and we have several opportunities to perform genetic studies. Even last time we were very lucky because we applied in a migraine field, and we got again a new support which is related to the so-called Eurohead pain, together with colleagues from Sweden, England, and Germany. We have a joint activities concerning the pathomechanism of migraine and to investigate some potential drugs which could help the treatment of this disorder. Also, we have an experimental neurology and behavior pharmacology laboratory. We have the chance to investigate open field, rotarod, eye maze, water maze, radial maze, which has good chance to investigate this compound. How do they influence the neurological, cognitive, and other parameters of the animals? And we have also a neuroimaging group. It's very close collaboration with the clinic. It's absolutely a clinical activity, and it is focused on multiple sclerosis, on migraine, and even on Parkinson's disease. And now we have a very close collaboration with Franz Fazekas. I will just meet within the next week, and we want to set up a very close collaboration also with the Graz University. So in conclusion, these are all research activities, and my special privilege to invite our students to say some words about the kynurenine system. You see, this story started at the Mass General Hospital, Boston, around 20 years ago, when Joseph Márton, Flint Beale, they found that there is an alteration in the kynurenine pathway in the brain, and our students, together with Dénes, was able to set up a method to measure kynurenine and its metabolites. So may I ask you just to start your presentation. Please, Márton.
The topic of my presentation is measuring concentrations of molecules formed via the tryptophan metabolism in different brain regions of mice and the validation of methods used during these assessments. Amino acid tryptophan is a precursor for many compounds in the different organisms. About 5% of the total amount are converted to serotonin, and the residual 95% go through the kynurenine pathway. As a very first step, L-kynurenine is formed from the molecule tryptophan through an intermediate molecule called N-formyl L-kynurenine. This step has a central role in this pathway. At this point, different ways are available for kynurenine to form its metabolites. One of them is kynureninic acid, which is known to be a neuroprotective agent, and the other one is 3-hydroxykynurenine, which has neurotoxic effects. The figure on the right side is derived from an article published by the colleagues of our clinic and their collaboration partners. Listed here, we can see some important neurological disorders which are related to the altered concentrations of the above-mentioned molecules. Such diseases are, for example, cerebral ischemia, Alzheimer's disease, Parkinson's disease, and Huntington's disease. These diseases, especially the first two mentioned, so in our case, cerebral ischemia and Alzheimer's disease, cause significant socioeconomic problems due to their high prevalence worldwide. Performing proper measurements in connection with the animal models of these diseases is made difficult by the fact that only a little amount of data is available in the literature, which are regio-specific and normalized. Furthermore, the related methods are usually not validated. Additional problems are caused by the various weights of the different brain regions and the consequentially inevitable normalization. Therefore, our aim was to determine concentrations of tryptophan and its free metabolites in the different brain regions of mice and to validate the applied HPLC methods by testing their selectivity, precision, recovery, and we also defined the limit of detection and the lower limit of quantification values. An additional aim was the application of reliable inner standards in order to detect the exact original concentrations. For our experiments, we used 20 weeks old C57 black 6 male mice. As a first step, the animals were anesthetized, and then thoracotomy was performed, which was followed by taking blood samples from the right ventricle needed for the parallel serum measurements. Afterwards, we performed perfusion through the left ventricle with artificial cerebrospinal fluid. The next step was the decapitation and then the isolation and dissection of brains on cool surface. Brain samples obtained this way were then stored at minus 80 degrees of Celsius till the next step of the process. Afterwards, these samples were homogenized by a sonicator in a homogenization solution of 250 microliters, which solution has already contained our inner standards. The homogenized samples were then centrifuged, and then the supernatants were removed, which were directly measured after this process, or if not, we store them at minus 80 degrees of Celsius till the measurement. For analysis, we applied reverse phase, high-performance liquid chromatography, IDEST HPLC. The detailed parameters of this method are listed on the left side of this slide. Kynurenine and one of the inner standards, pre-NLT, were measured by UV detector, and kynurenic acid and tryptophan were measured by fluorescent light detector simultaneously. In case of free hydroxykynurenine, we used a different method. Concentrations of free hydroxykynurenine and the corresponding inner standard isoproterenol were measured by electrochemical detection. In order to perform the most accurate measurements, we plotted the voltage current curves of these molecules, and we also determined the optimal working potential, which is the minimum voltage value belonging to the plateau phase of the free hydroxykynurenine curve. In our case, this value was around 650 millivolts, as we can see on this picture. Under such conditions, we can measure peaks with good detectability for both free hydroxykynurenine and isoproterenol, along with a properly low level of noise. In this figure, chromatograms derived from our measurements are presented, where I would like to demonstrate our system selectivity. In the first column, chromatograms can be seen derived from our kynurenine and free NLT measurements. In the second column, we can see chromatograms derived from our kynurenic acid and tryptophan measurements. And in the third one, we can see chromatograms representing free hydroxykynurenine and isoproterenol measurements. The first column represents standard samples. The second one represents native biological samples, and the third one represents spiked biological samples, which means that known amount of our metabolites and inner standards were added to these originally native samples. In the native biological pooled samples, kynurenine has a good detectability, and there is no disturbing noise at the retention time of free NLT. Furthermore, as the chromatogram of the spiked sample indicates, we have great selectivity for both of these metabolites, and we can identify the peaks by their retention times reliably. The same results can be seen at the measurements of the kynurenic acid and tryptophan. Regarding free hydroxykynurenine measurements, in the native biological pooled samples, free hydroxykynurenine concentrations were measured around the LOD. However, the results of the spiked sample indicates great selectivity for this molecule as well. Elevated levels of free hydroxykynurenine are expected in brain samples derived from mice which suffer in some neurological disorders or are treated with some special toxins, and that concentration range would be proper for us to perform valid measurements with this method. Our further validation results can be seen in this table. Our validation results correspond to the principles of the International Congress on Harmonization in every instance. Robustness of our method is weakened by only one value, which is the relative standard deviation of precision of concentration of free hydroxykynurenine measurements, which slightly exceeds the optimal 5% value. On the bottom of this slide, we can see plots illustrating the correlation between the concentrations of our inner standards and the weights of the brain samples. To define the distribution of the weights of the brain samples, we use Shapiro-Wilk test. According to the results, the weights of the brain samples did not show normal distribution. Therefore, for our further data analysis, we applied the non-parametric Spearman correlation. As we can see, both inner standards concentrations show adequate correlation to the weights of the brain samples, along with a properly high level of confidence. However, in case of free NLT, the results were more reliable. Still, the application of isoproterenol is necessary because of the different settings methods, since free NLT cannot be detected with the applied method of electrochemical detection. In this table, I listed the median values of our results compared to the reference data available in the literature. As indicated, these informations are specific to striatum, cortex, hippocampus, cerebellum, and brainstem, and are related to the above-mentioned 4 compounds. White numbers represent validated data. As we can see, our results are in the same concentration range as those values which were validated in the literature. Pre-hydroxyquinoline concentrations were measured around LOD in the native samples derived from mice, which were healthy and were in our control groups, as well as in most of the literature data. However, as I mentioned earlier, due to its toxic effects, we expect elevated and well-detectable concentrations of this molecule in our future projects of experimental groups. We did not find such a publication which contained data from all of the above-mentioned 5 brain regions, and the related methods were rarely validated. Thus, our future research results could possess novelty. Based on these results, we can conclude that we have a reliable and robust method which is applicable for further valid measurements in connection with the tryptophan metabolism in animal models of the different neurological disorders. Inner standards with good quality are available for data normalization, and there is a possibility for publication new results in this field. Finally, I would like to say thanks to my colleagues who take part in this job for their hard work, and last but not least, I would like to say thank you for your attention.